Hi, everyone. Thank you so much for being here at seminar today. I want to remind you that there's a social hour immediately after the seminar in the Mulford Courtyard. So today it's my great pleasure to introduce our speaker, Dr. Racine Rangel. Dr. Rangel is a UC President's Postdoctoral Fellow and a Climate Justice Fellow through the Center for Reimagining Leadership at UCSC. Dr. Rangel's research sits at the intersection of ecology, physiology, nutrition, aquaculture, conservation, and management, and investigates how individual organisms, populations, and communities respond to global change stressors on the California coast and beyond. Dr. Rangel's work also tests anticipatory management strategies that could be used to future-proof populations of critical marine species. And her work spans a number of economically valuable vertebrate and invertebrate species and is conducted in collaboration with local stakeholders to ensure that her findings are relevant to local sustainable management of natural resources. Dr. Rangel has amassed an impressive number of honors, awards, and research grants during her academic career, which includes a Bachelor's of Science in Biological Sciences at the University of Nebraska, 2 master's degrees, one from UC Irvine and one from Cal State U Long Beach, and most recently her 2023 PhD in Ecology and Evolutionary Biology from UC Irvine. In addition to her published works, which have received awards such as Best Paper and leverage manipulative field experiments and lab-based studies, Dr. Rangel has made important contributions working with aquaculture through a Minorities in Aquaculture internship, as a teaching excellence and innovation pedagogical scholar, through which she designed a pedagogy program and led it for GSI's multiple years. And she's conducted impactful outreach as a writer and managing editor for NPR's Lowdown on Science program. And she serves as a mentor in at least 4 that I counted mentoring programs for underserved groups in STEM. So Dr. Rangel, thank you for being here and sharing your work with us.
Wonderful. Thank you so much for that introduction, Adrienne. And thank you all so much for being here today. I'm really excited to share with you some of my past research and current and future work. So without further hesitation, let's get into it. So here we can see a wide array of different ways of how we as humans derive resources from the sea, right? And as a physiological ecologist, I'm really interested in the overarching question of how can organismal physiology here inform adaptive management? And specifically, I'm interested in the overarching question or thinking of how individual physiological processes can scale through levels of organization, through populations to communities to ecosystem services, thus changing the way that we as humans interact and manage those resources. And specifically, I'm interested in how individual physiological processes can also directly influence ecosystem dynamics, and what does that look like for adaptive management? So let's, when I'm talking about adaptive management here, I'm also including the notion of sustainable aquaculture. And so let's look at this artist's rendition of a kelp farm up north in the state of Washington, Pacific Sea Farms. And so here we can see that kelp is being grown on these ropes, and then within and in between that farm, there's also the co-culturing of shellfish as well. And we can see that this is occurring within a naturally dynamic system, right, in a coastal system. And so we know that individuals within this system are interacting with their environment. So let's focus in on this individual fish that we see here. Well, we know that individuals need to consume some type of food, and then that food is then allocated to different physiological processes, right? Things like metabolism, growth, reproduction. And when we're thinking about shellfish or ectotherms, like the fish that you see pictured here, we know that these physiological processes are closely tied to the environmental conditions that individuals are in. So now we have an idea of how this individual is interacting with their environment, but we know individuals don't exist in a vacuum, right? There are so many other things occurring with natural variation within these systems. And so you can have interactions of this individual with individuals of the same species, making up population level interactions, as well as interactions with individuals of different species, making up community level interactions. And I really want to stress here that within this network, right, we as humans are also now deeply embedded within these systems and these interactions, right? We are changing coastlines, we are creating farms that are altering coastal ecosystem functioning, and the services and the goods and resources that we receive from the ocean. And so I'm going to talk briefly and touch on my path of kind of how I've been building my skills to answer my overarching question. So as Adrienne pointed out, I grew up nowhere near an ocean in the middle of the country. I grew up in Nebraska, and I started studying this little fish that you see here, the fathead minnow, and I was looking at the aquatic toxicology and endocrinology of this species. And then I didn't really know what grad school was. So after undergrad, I was lucky enough to land a technician position working for Dan Bolnick at UT Austin before he moved to the University of Connecticut. However, I actually didn't move to Austin. I moved straight to Vancouver Island, where I was living for the first stint of my job working as a tech for him. And here I started getting interested in being able to build skills understanding the ecology and evolution of fishes, and particularly the three-spine stickleback in glacial lakes. And so we just published another paper in this system last year. However, I won't have time to get into that today, but I'm always happy to chat with people about stickles. And so after my time teching with Dan, I really had wonderful mentorship in his lab and felt like I was ready to start grad school and begin my journey. And so that brought me to Southern California, where I started my master's at Cal State Long Beach, working with Darren Johnson. And here I started to really build out my skill set of combining physiology and ecology. And I started studying different benthic nearshore fishes that you see here, the blue-banded goby and the woolly sculpin. Again, I'm not going to have time to talk about this, but I love discussing these really unique, wonderful fishes. And so happy to discuss that at a later time. And so then I decided, you know what, grad school is cool. Let's do more grad school. And so I started my PhD down at UC Irvine, but I have a really hard time staying in a space for really long periods of time. And so actually all of my PhD research was done up in Southeast Alaska, which I'm going to talk about today. And during this time, this is where I really started building and starting scaling and thinking about how individual physiological processes scale through levels of organization and thinking about ecosystem function and services in coastal systems. And so that leads me now to my current adventure as a postdoc at UC Santa Cruz, where now I'm starting to build and pull together ecology, physiology, and aquaculture, looking at this in red abalone in California. So of course, throughout my journey, I have learned that there is a great challenge to answering my overarching question, right? And that's because there are multiple ways in which physiology can interact and alter ecosystem dynamics. And so I'm going to walk you through my framework of kind of how I'm thinking about tackling this question. So we'll start with the idea of environmental conditions, and those directly influence ectothermic species, right, or organismal physiology. That can then scale to influence community level interactions and assemblages and structures, leading to shifts or changes in ecosystem level responses like ecosystem functions. And then that scales to influence how we as humans manage and adaptively manage these resources and species, right? And of course, like all good scientists, we know it's never this straightforward. In fact, it's really intense, and there's a lot of moving pieces here. And that includes things like temporal variation, right? There's seasonality of these environmental conditions, altering and changing the pathways within this framework, as well as resource variation. It's very important, right? The quality of food, the type of food individuals have access to can all shift how this framework is interacting. And throughout my career, I've really been interested in focusing and trying to understand how does climate change now drive alterations within this framework? What does that mean for the species that we manage and collect and harvest from the sea? So today I'm going to be talking about a couple different stories and projects. The first one's going to talk about incorporating a wide array of different variation that you see here of manipulations in situ. And then the next project that I'll talk about today is then thinking about how resource variation fits within this framework and thinking about adaptively managing those resources. And so first, let's talk a little bit about how climate change is altering our marine systems. So here this figure is showing you CO2 emissions across our globe. right? And our oceans have been doing us quite a huge favor for quite some time now by absorbing about 90% of the excess heat from these CO2 emissions, as well as about 30% of those CO2 emissions. So what does that actually mean for our marine ecosystems? Well, we know one of the most prominent changes we're seeing is changes in ocean warming, right? So here this figure is showing you changes in sea surface temperature from 1901 to 2020, and every single black dot that you see there is a significant increase in sea surface temperature during that time. And importantly, climate models are predicting a further increase in sea surface temperature of about 1.7 degrees by the year 2100. We also know another consequence of those CO2 emissions is that our oceans are becoming more acidic, right? We have ocean acidification taking place. So here this figure is showing you the CO2 time series in the North Pacific with CO2 emissions on that left y-axis, pH on the right y-axis, and then year on the x. And that top red line is showing you the increase of CO2 emissions, right? And then that second green line is showing you the consequence of our oceans absorbing those excess CO2 emissions. And then that bottom blue line is showing you the subsequent chemical reaction that's taking place, leading to a decrease in ocean pH over time. And so climate models are predicting a further decrease of about 1.6 pH units by the year 2100. And so now looking and thinking, we have an idea now, right, of how climate change is influencing this framework and the two drivers that I'm going to be talking about today. So let's talk about the first project. So here we're going to be looking at this in mussels. And the question that I'm trying to answer is, how does warming and acidification impact coastal ecosystem functioning? So let's talk a little bit about the intertidal system, right? So here's a really awesome artist rendition of Bob Payne's work, which was all done up in Washington State. And Bob Payne was one of the pioneers within intertidal ecology that really showcased the power that we can use the intertidal system as a tool in understanding ecological theories and shifts in community composition. One, because he would go out and he would collect sea stars and he would take them off the rocks and he'd chuck them as far as he could into the ocean. And he just did this, he removed all the sea stars. But by doing this and manipulating this environment, he was able to showcase the importance of sea stars being a keystone species. And so building off of other titans in this field, like Bob Payne and others, using the intertidal as a system, we can manipulate these environments to test these ecological theories, looking at shifts in communities. Importantly, we can track populations over long periods of time, right? And really see how climate drivers are altering populations within this system. And then we can use it as a really powerful tool to scale up, tracking individuals, and then looking at how we can look through that through populations, community, to then testing ecosystem functions as well. So using this system, my study system is in Sitka, Alaska. You can see pictured here, it's a really beautiful place to work, and it's located at a site called John Brown's Beach. And so this is during low tide. So the water is out, creating little pools of water that get stuck within the rocks. And you can see that there's a lot of different species living within these pools. And then during high tide, all of those pools get covered again with water. And so myself and colleagues, we set out to test the effects of climate change on this ecosystem by conducting a 6-month-long factorial field manipulation. And so we had 20 different pools that we were studying, and we had 4 different treatments. So we had pools that were unmanipulated, pools that were warmed, pools that had CO2 added to them, and then pools that had CO2 added plus were warmed. And the color scheme that you see here is going to remain consistent for the figures in this project. So I really hope you're trying to picture to yourself right now, like what that looks like in an intertidal system. And it required a ton of hardware. And so this is one of our pools that had CO2 added plus was warmed. And so how we powered this was we had these batteries, which were 9-pound motorcycle batteries that were in these waterproof cases, and they powered our heater bars, which were anchored into the bottom of the tide pools. And then we had this little yeast reactor here, CO2 generator. So we used baking yeast that you use to make bread, mix it with sugar, bicarbonate, some warm water in a Gatorade container. Figured out that was the perfect amount. You shake it up, you put it inside of this little otter box, And then the yeast consume the sugar, creating CO2, creating a little microbrewery in there. And then it pumps CO2 through an airline and air stone into our tide pools, thus decreasing the pH. So here's a close-up video you can see. So this is the CO2 bubbling out of that air stone into our tide pools. And then here's a close-up of what one of those heater bars look like anchored into our pools. And so, of course, you can imagine this was a massive effort to do. So this was a part of a much larger collaborative effort. And so I want to give a shout out to my co-authors here at UC Irvine, Santa Cruz, and San Diego State University. And my part of this research was I was really interested in thinking of how these climate change manipulations are influencing mussels that you see pictured here in that yellow circle. And specifically the Pacific blue mussel or Middleus trosselis. And I really wanted to understand these climate change manipulations on these mussel populations. One, because they're foundation species. So they're really important in these systems. They create habitat and increase biodiversity for other species in this system. And if we look at the breakdown of the sessile communities, it's also the most abundant sessile animal and the most abundant calcifying species. So we're thinking impacts of acidification. Species that build with calcium carbonate may be more negatively impacted by acidification. So mussels seems like a really wonderful species to focus in on for these manipulations. So let's talk a little bit about the potential effects of acidification and warming on organisms. So focusing in on mussels here, potential ways that we could see these drivers altering them could be an increase in metabolism, an increase in energy demands, or an increase in growth. And when we're thinking about calcification, right, we're likely going to see decreases in their ability to make and repair their shells. So you can see the difference between these two mussel shells, where the top one was in an ambient pH condition versus this lower one, which was clearly held at low acidity or high acidity conditions, thus impacting its ability to repair its shell over time. And I really want to highlight here the importance of food, right? So some really awesome work done by Melsner et al.'s group in Germany showcased how having access to high amounts of food and that resource variation can actually offset those negative impacts of acidification on calcifying species like mussels. And what about potential effects of acidification and warming on populations? Well, sticking with our mussel theme here, we actually saw the effects of warming just a few years ago along our Pacific coastline, where marine heat waves moved through and just decimated populations of mussels along our coastline. And so we saw big changes and decreases in survival and population abundance, which then leads to shifts in community interactions and structure and composition as well. And then when we're thinking about ecosystem function within these coastal ecosystems and in tide pools, mussels again play a really important role because they're dominant calcifiers in this system. And so they maintain an ecosystem service known as net ecosystem calcification, or NEC, through that process of building their shells, right, through calcification. And so net ecosystem calcification is just the rate at which calcium carbonate is transformed from its aqueous form to its solid form, the building of those shells. And so if a system has positive values, it's likely in net calcification. So that would mean individuals are able to grow, they're able to build their shells, get bigger, right? Where if a system is in net dissolution or has negative values of net ecosystem calcification, that would mean dissolution's likely taking place. So you would expect to see mussels that have, maybe they're not as big, or their shells are actually corroding, and that the calcium carbonate is moving back into that aqueous form. So let's look at how our climate change manipulations were influencing ecosystem function here. So we have net ecosystem calcification on the y-axis, and then time in months on the x-axis. And so more positive values mean net calcification is likely occurring, more negative values mean net dissolutions occurring. And this figure is going to show you the difference between the day and nighttime values, and then the treatments are color-coded there in the legend. So let's look first at March. So this is pre-manipulation. This is before we turned on any of our manipulations in our pools. And really what I want you to take away from here is that net calcification increased during daytime tides, okay? And then it was more negative dissolution at night, during nighttime, okay? So now that dotted line is indicated, we are turning on our manipulations. And so looking at our first time point after that in May, we see this same trend, except it's bigger. And that's likely due to the fact that we are now moving into growing season, right? So water temperature is now no longer freezing. Things are starting to wake up. Water temperature is starting to get warmer. And so individuals are starting to grow. They got to take advantage of the warmer waters, and they're growing, they're calcifying more. So we're still seeing that same trend of net calcification increased during the daytime tides. And then we start to enter our hottest months in Sitka. Okay, and we start to see an interesting pattern here. And I want to highlight this by these yellow boxes here, looking at those CO2 added pools, where here we saw that CO2 pools were exhibiting dissolution both during daytime and nighttime tides, right? And this is also occurring during those hottest months. So an already added stressor on top of having low or CO2 added to those pools as well. And then we start entering August, water temperatures starting to kind of cool down again, and then things are kind of starting to go back to normal and following that trend again, where we're looking at more calcification during daytime tides. And so this is an overview of how our net ecosystem calcification was being influenced by these manipulations. Let's take a look at how community structure was being altered. So here's an NMDS plot showing you the sessile community structure within our pool. And what we found is that treatment was a significant effect with our CO2 added pools being different from our warmed pools. But I really want to highlight here that what we're seeing were these minor changes in community structure. One, we didn't see any mass die-offs of mussels during our experiment. And just randomly, we tended to have more mussels in our CO2 added pools versus our warmed pools. And so this is really highlighting that there's any shift in community structure is not accounting for any reduction in net ecosystem calcification, because the mussels are still there, right? We didn't lose large sections of mussels. And so this shift is likely just due to our warming pools also having high amounts of seaweed species present in them, just randomly the pools that we picked. So no big shifts in community structure. So let's look at how individual physiology of the mussels were being influenced by these treatments. So now we have mass specific oxygen consumption, or MO2, as a proxy for metabolic rate on the y-axis, and then our treatments on the x-axis. And so what I found was that in those CO2 added pools, individuals had an increase in metabolic demands, right? And so this is likely suggesting that there's a physiological cost of being within these high CO2 environments. And so what does this look like when we're looking or thinking about individual physiology directly influencing ecosystem function? So now we're looking at net ecosystem calcification on the y-axis and MO2 of the mussels, individual mussels on the x-axis. And I'm only going to be talking about the daytime tides and data right now. So what we expected to see and what we found is highlighted by these solid arrows that you see here. And it was as metabolic rate increased, net ecosystem calcification increased under ambient CO2 levels. And so these are our unmanipulated and warmed pools, right? So you have individuals that are growing more, they're calcifying more. You should have an increase in net ecosystem calcification, and that's going to cost more metabolically, right? But then we saw an interesting turn when we added CO2 to these tide pools, where we saw net ecosystem calcification actually decreased under CO2 added conditions. And this is highlighted by these dashed arrows now that you see. And you can see that really highlighted by those CO2 added pools at that bottom dashed line, where here this is likely highlighting that individuals were under chronic stress. So they had higher metabolic rates, but they're not calcifying the same. And so we're actually seeing a decrease in net ecosystem calcification in our pools. And so let's look now at how our populations of mussels are looking. And as a reminder, right, we didn't see any big shifts in population or community structure of our tide pools, so we didn't lose lots of amounts of mussels. So now we have net ecosystem calcification again on that y-axis, and then mussel percent cover, or like abundance of mussels on that x-axis. And so what we found, what I expected to see, was that as mussel abundance increased, net ecosystem calcification also increased under ambient CO2 levels, again highlighted by those solid arrows that you see there. There were more mussels in the pool, there was more calcification taking place. But then we saw another interesting trend when we added CO2, where now net ecosystem calcification is actually decreasing under CO2 added conditions, right? And we can really see this in our CO2 added pools, which just had higher amounts of mussels randomly. But we can really see that regardless of having high amounts of calcifiers in those tide pools, we're still seeing a decrease in net ecosystem calcification. And so I really think this is highlighting again that importance of individual physiology and those processes of having high metabolic rate, but not being able to calcify, right? And keep up with being in this high CO2 environment. And so from this project and this study, I found that changes on ecosystem function are occurring through populations, but it's really that per capita, that individual physiology that's really driving those shifts that we're seeing in these tide pools under these manipulations. And I really want to stress here that we would likely see continued changes in community structure over longer timescales if we ran this for even longer than 6 months, right? Where as those individual physiology thresholds are surpassed, we'd start seeing sharp declines in survival and population abundance of mussels, thus then shifting community structure rapidly. And so implications of this system and this study, right, is really being able to manipulate these systems, these tide pools, and allowing us to incorporate all the variation that exists in these systems, but looking and seeing these strong emergent effects of these climate change drivers, and we can do that in the field, right? As well as determining vulnerability in ecosystem services and thinking, what does this look like now for other valuable ecosystem engineers? Things like oysters, for example. And so that's the kind of the wrap up of this initial first project. However, this was, I was finishing my PhD and I knew that I wanted to continue working within this framework, but I wanted to start adding in, again, that adaptive management. How are we as humans interacting within this framework? And what does that kind of look like? And so that luckily led me near the end of my PhD to doing an internship with Minorities in Aquaculture, or MIA. And so this nonprofit was founded and created by Amani Black while she was actually a grad student. And the whole goal and purpose of it is to increase diversity within the blue economy, right? And so I had the immense privilege of being able to go and work on predominantly women-owned oyster farms in Maine for a summer. And one, I really just wanted to know what it was like working on an oyster farm. Every farm does it different. Every farm has learned things differently. The knowledge that they hold is different, just in the same bay, but farmers do things differently. But I also wanted to talk and understand their needs, right? How is climate change driving alterations within their farm now? What are solutions that they're looking for from researchers? And so this working in this system and working with these farmers really allowed me to kind of apply this to my research framework of now combining aquaculture, ecology, and physiology. And so that really brings me to my journey now of working with abalone of the West Coast. And if you don't know what an abalone is, that's okay. They're a very large marine mollusk or a large marine snail. And they're really awesome animals. And they, along our coastline of California, the West Coast, we have 7 different species that reside in our waters. And you can see that pictured there on the right side, their historic ranges in California. But really, I want to stress that abalone have been loved and treasured by indigenous peoples for thousands of years along the West Coast. And so here are some black abalone you can see attached to rocks during low tide at the Channel Islands. And they basically become a rock on top of a rock during low tide, but they're also moving around and eating. And so we've seen effects of how indigenous people have worked with abalone by harvesting them. So here's a picture showing you a shell midden on San Nicolas Island. So this midden's about 6,000 years old. So abalone were being harvested from our coastlines for a very long time for food. But as well as on top of food, people have long been mesmerized by their beautiful shells, right? The inner nacre, this beautiful shiny shell that you see pictured here. It's on the inside, right? So once you remove the meat of the abalone, this beautiful shell exists here. And so these shells became very important, right? They were utilized in making tools, things like fish hooks that you see here that were found at Santa Barbara Island. They were used as a form of currency and trading, right? These beautiful abalone shells and really big abalone shells from red abalone. And they're also used in ceremonies and for regalia, right? So things like jewelry. This is highlighted by this piece that you see here by a Haida wolf pendant created by Haida artist Phil Reed, where you can see that the abalone shell has been broken and cut and then really laid delicately within this jewelry piece. And so for so many years, humans have enjoyed and loved abalone along our coastline. And so much in fact, to the point that we almost love them to death. And so this is a picture from the 1930s in Morro Bay, where we were harvesting so many abalone and loving them and eating them so much that we were creating mounds that people could literally lay on top of. And this was outside of a restaurant in Morro Bay. So that is the amount of abalone shells that they were serving to people at this restaurant during this time. And so, of course, we started to see rapid declines in combined species of abalone starting in the late 1960s, moving into the early 1980s. And so much so that by 1997, fishing for all abalone species closed, essentially ending all commercial fishing of abalone in California. And so abalone live within the intertidal, they live subtidally in kelp forests that you see pictured here. And we kept seeing declines, right? We were seeing less and less adult abalone along our coastline. So much so, these declines in health, low recruitment rates, density of adults, that the white abalone became the first marine invertebrate to be listed on the Endangered Species Act in 2001, followed by the black abalone in 2009. And then the red abalone was kind of like the last abalone standing, right? There was still a recreational fishery open for this species. However, in 2018, this fishery also closed with no plans of reopening it as of right now, right? So now we have essentially lost all ways in which we can collect and harvest abalone from the wild along our California coastline. So you may be wondering, why was this occurring? What's happening? And it's really a perfect storm of a lot of things, but one of them being changes in abalone food sources. So here this figure is showing you kelp cover in Northern California in Sonoma and Mendocino County. And you can see in 2008 there, that first line, you can see all the kelp cover, right? We had really healthy populations of macrocystis kelp and bull kelp along our coastlines. And then in 2014, we had one of the largest marine heat waves move in, and it stayed for a really long time, becoming the infamous the blob. And it just decimated populations of our kelp along our coastline. And that still remains today. We haven't seen our kelp populations really return back to normal. And so really it's this combination of loss of abalone food, changes in environmental conditions, disease as well as those harvesting pressures of abalone that led to these decrease in all of our 7 abalone species on our coastline. And of course, I really want to highlight here that researchers were on the ball. They were trying to understand, how do we find solutions for abalone along our coastline? And so one of my collaborators, Dan Sweezy, who's located at Bodega Marine Lab, did a really awesome study during his PhD, where he highlighted the importance of paternal or maternal investment in the next generation of abalone. So under high CO2 conditions, lipid provisioning from the parent to the larval offspring was a really strong predictor of survival in abalone. And you can see that highlighted on the right side of this figure with the high CO2. So this is really highlighting a mechanism potentially of thinking about how food of parents kind of play within this framework, and what does that look like when we're thinking about restoration of this species in the future under a changing climate. And so I want to stress here that right now in California, we're at this really interesting crossroad where we are relying solely on aquaculture for obtaining red abalone in California for food, as well as thinking about restoration efforts. And so these include farms like the Cultured Abalone Farm down outside of Santa Barbara, as well as the Monterey Abalone Company in Monterey Bay, that you see how they also grow their abalone very differently between the farms. And I want to highlight here that thinking of these adaptive management and solutions for abalone is going to require very collaborative and creative and anticipatory strategies across many different interested parties. And we're already seeing this, right, across state lines for many different species. So that includes things like kelp farming up in Alaska or oyster farming in Washington. And Washington is also kind of showing and highlighting the importance of using sustainable aquaculture to then create pinto abalone recovery plans, growing individuals in a hatchery in an aquaculture setting, and then releasing them back into the wild. As well as this goes across borders internationally, right? And so I had the immense privilege of being able to attend the Haida Gwaii Abalone Summit, where we were really focusing on northern abalone or pinto abalone, but really looking and understanding and listening to all these different interested parties that really just at the core of it loved interacting with the ocean, with the resources that we as humans can obtain from them, and for food as well as for cultural reasons as well. And we see this on our coastline in California with the Kashaya Band of Homo Indians who are actively starting to create and try and build an abalone aquaculture farm within California. And so this brings me to my framework here for this last project I'm going to touch on, where we're including resource variation and thinking about adaptive management, but also adding to this framework and thinking about how plasticity and adaptation interactions are also going to reshape this framework moving forward. And so for this project, I'm focusing on red abalone, and I'm asking two questions. One being, does diet influence red abalone investment under high CO2 conditions? And then can we identify adaptive strategies for more climate resilient abalone in the future? And this is a part of a much larger collaborative effort. And so this myself and Christy at UC Santa Cruz and working with abalone farmers down outside of Santa Barbara, as well as all the abalone experts at UC Davis's Bodega Marine Lab. And so for this project, we're looking at two different types of seaweed here for our diet treatments, one being this beautiful red ogo seaweed, and the other being another red or dulse seaweed. So both red seaweeds, but they look vastly different, which is really helpful when growing and feeding them out. And so abalone have been feeding on either 100% ogo, 100% dulse, and then a mixed or a combo diet, 50-50 of these two species. Here's a sneak peek of what that looks like growing it at our mesocosm system at Santa Cruz. We use what's called a tumble culture, allowing them to photosynthesize outside and giving them nutrients. So we've been growing our own seaweed at Santa Cruz. And then we have our abalone, which have been housed at either in a low CO2 environment or a high CO2 environment and being fed these different diet treatments. And then throughout this experiment, I measured a whole slew of different things like growth, grazing, digestion, efficiency, efficacy, respiration, survival, and we just finished this experiment. So from start to finish, we ran this for almost 9 months, so 8 months. It was an 8-month-long experiment. You can see that the abalone were housed in these condos here, and then they were fed their different diet treatments. So I'm going to show you some preliminary data from this. I just finished analyzing it, so it's likely to change at some point, but I wanted to show you some of our preliminary results because we're seeing some interesting patterns. So here we're going to be looking at growth rate of our abalone, and then on the y-axis, and then the different diet treatments on the x, and then we'll be looking at our low CO2 versus our high CO2 pH treatments. So first what I found was that red abalone had increased growth rates on the dulse and mixed diet. So looking at ogo first, we can see here that there's no difference between our pH treatments, right, of how much they were growing on those diets. But then comparing that to our dulse treatment, now we see that there is a higher amount of growth that occurred on this dulse. This is 100% dulse. And the fact that now there's an effective pH, so that low CO2 abalone on this dulse diet grew significantly more than our other treatments. And then we can look at our mixed diet here, which is now kind of falling in between our ogo and our dulse, but again, now not seeing any strong effects of pH. So now let's see look at how the abalone were actually grazing or eating on these different diet treatments. So the main point that I found was that grazing rates were actually higher for the mixed and Ogo diet, even though they grew more on the dulse diet. So looking at Ogo here, we can see again, no difference between our pH treatments. But now with our dulse treatment, we see that they were actually consuming less of the dulse or grazing less on the dulse treatment, but they grew significantly more on what they were eating. And then the mixed diet is again kind of falling in between, but a little bit higher now, where they were really eating a lot of the mixed diet, but again, no strong effect between the pH treatments so far, but we are seeing this difference in the amount that they're grazing and the amount that they're growing on these different diet treatments. So projected outcomes from this project, I really am hoping to detect effects of individual resource variation on physiological rates under varying pH, as well as thinking about how this research can be applied to aid restoration efforts with different diet options, and really highlighting this importance of maternal investment, because we're not seeing any strong effects of pH as of right now, but we know that diet is likely going to play an important role here in creating more climate resilient offspring of abalone in the future. And so looking forward, I'm really excited to continue adding to this framework. One of the ways that I want to do this is by still conducting these long-term field manipulations, right? Utilizing this intertidal system for us to be able to look at these shifts in ecological communities along coastlines. What does that mean for shifts in ecosystem functions and services under climate change? As well as continuing to work within this realm of resource variation and adaptive management, sustainable aquaculture. And really, I'm excited to continue working and having abalone be a really key species within this framework, but also adding in other species, right? Things like scallops or urchins, as well as looking specifically at things like seaweed farming and how different species of macroalgae can be utilized within this framework, and thinking about whole culturing of species along coastlines moving forward. So I'm going to continue and look forward to answering and trying to answer my overarching question of how does organismal physiology inform adaptive management moving forward. But a really big part of my research is that I really aim to increase the recruitment and retention of underrepresented individuals within STEM, and particularly within the marine science field. And so I do this by utilizing mainly four different approaches, one being having student-centered mentoring. So I still hope you realized or noticed my students who helped on some of these projects and highlight their immense, wonderful efforts of, these projects take a lot of work, and they're really rock stars helping me get all of this research done, as well as including inclusive and experiential teaching, right? I've had the immense privilege of being able to be at institutions that care a lot about teaching. And we know having access to hands-on experiences, things like field courses, are instrumental in increasing the recruitment, but also the retention of underrepresented students while wanting to be within STEM fields, as well as increasing identity and accessibility and inclusivity. So utilizing the networks that I've been growing with partners like Minorities in Aquaculture or the Women of Color in EEB group, right? This also includes mentoring efforts and thinking about, utilizing my own identity to help students create that sense of belonging within the STEM field and creating opportunities. So I think it's really important to think about ourselves in academia of, recruiting and retaining individuals from underrepresented backgrounds starts long before they're in a college classroom, right? So utilizing outreach and service as a way to start recruiting them really young. So utilizing things like the Seymour Marine Discovery Center, which is down at UC Santa Cruz, or really awesome groups like Girls Inc., which are in local communities. And really a big piece of this is opportunities when we're thinking about marine science or field work, right? It's very expensive to be in this field. Scuba diving equipment costs so much money. I've lived through this where during my master's, I was diving a lot for my research. And I had to decide many times between getting my gear serviced and diving safely or buying groceries and paying rent. And so students are still dealing with this today. And so making sure that, as we're moving forward, we're creating pockets of money and having that funding be at the forefront of our work, and creating, systems that allow students to not have those financial burdens throughout their time in the sciences. And so I just want to give a shout out to my collaborators and acknowledgements. This research, all of this research, just takes a team, a mountain of people to conduct. And so there's so many, I could talk for probably another 40 minutes of how much I love all of these people and appreciate their help, as well as all the funding sources that have trusted me to do my research and then report back to them. And with that, I will say thank you, and I hope I have some time for questions.
Thank you so much. That was just wonderful. Great. So in the first case study, I might have missed this, but the pools, the heating and the CO2 are in the pools when the pools are isolated, but then the tide comes in and everything gets
Turns off.
So how does that play a role? It's not the day-night thing, like how?
No. So that's what I think is a really cool thing of this project. And really what I was interested in seeing is that essentially the system resets every 12 hours when the tide comes back in. So all these treatments were programmed to turn on during low tide, and then they turn off during high tide. And so we're still seeing these super strong emergent effects with that reset every single day, with fresh seawater coming in and flushing all of our treatments out.
So the assumption is that in the natural setting, it'll be even more exacerbated, the effect or the impact would be even more exacerbated because there won't be a reset.
Well, yeah, so if they in the future, when the starting pH is already much lower than it is now, yeah, you would expect that even with the reset, you're already starting at a much lower pH or a much higher temperature, right? And so this was kind of our way of projecting that out into the future right now. And we then looking and seeing how the community structures changing, or how individuals are being influenced by these manipulations. Yeah.
Do you have a nutritional profile of adults versus the old?
Yes. We I took samples throughout our experiment to test just that. So we'll hopefully be getting that data at some point. Just haven't had the time yet to work it up. But yes, also interested in seeing, can we figure out, is it more proteins? Is it more lipids? What is it about the difference between these two different seaweeds that clearly they're growing differently on them, and what does that mean, right? Yeah. Yes, yeah.
And I'm really curious as to sort of these pools. Do they, what pH would you expect pH to fluctuate in these particular pools?
A good question.
How does that, how would that kind of correspond to the particular?
Yeah, so this was it's a really wonderful question. So it can fluctuate a lot during a tide cycle. So in some pH or intertidal systems, we've seen shifts of like 7 pH units across the tide cycle. So the first year of this experiment, we just did a seasonal analysis and collection of water chemistry to kind of see if we can pinpoint how much that fluctuation is occurring. It's in nature, so the next summer we went out and it was just naturally hotter. And so we can't really get an idea of like what the baseline is, but we can utilize our unmanipulated pools to kind of look at that. And so the shifts weren't that much across our different tide pools. I think the biggest swing was maybe a 4 or 5 pH unit shift throughout a tide cycle. And that varied between day and nighttime as well. But really the important thing here is that we never saw it go below a I think it was like 7.5 was like the threshold, even at night. So I think that's really highlighting that, these communities within these tide pools, they have some buffering effect that's not allowing the pH to go super, super low where everything would just die. And then during the daytime, you have the seaweed communities, which can photosynthesize and kind of increase the pH during the daytime tides. And so we just allowed all of that to happen within the tide pools and just sampled throughout the entire tidal cycle to see, would we see strong effects of pH or temperature? Yeah. I think there was another, behind.
Yeah, for the Abalone bills, did you find that particle size of the seed is something a difference? I just know in oyster farming that was high.
They're like little lawnmowers, so no, not really. They're like garbage disposals, essentially. It looked like they were eating all of the time. Like I felt like I was just constantly feeding them. And so it didn't seem like there was a preference. The ones in the dose treatments were eating all of the dose that we were giving them, and the ones in the OGO treatments did seem like they were finishing their rations a little quicker. And I don't know if it's like the branching of the different seaweed, that maybe that's a preference. But really, I feel like abalone, if there's seaweed, they're gonna find it, and they're gonna eat it. So, yeah, but interesting I wish we could have also looked at macrocystis, which has the really long blades, comparing that to these kind of more branchy looking reds, but we didn't have the space to do that as another treatment. Unfortunately, maybe in the future.
Do you have any ideas to why in the first experiment, the CO2 plus warming didn't always follow the same?
Yes.
Pattern?
Yes, so I think so I have a whole other section of data on this where I was really diving into how the mussels were being influenced and their shells and the functionality of how their shells were changing under these treatments. And so throughout all of the data that we collected, those CO2 added plus warm treatments kept falling in between. They were never mostly as a negative of the CO2 added pools. And so I think that's highlighting that, our warming treatments didn't warm as much as we wanted. So there was like more of a moderate warming, and that seems to be offsetting those negative effects of acidification. So moderate warming to some extent may actually help offset negative effects of acidification within this system, which was not what I was expecting to see. I thought it would be much worse for the mussels in particular, but it seems like it falls in between. So something about the warming, and it might be that warmer temperatures allow more off-gassing to take place. So maybe the pH isn't as low. Yeah.
Did you see any kind of interspecific variation to your CO2 treatment?
For the mussels, like in their metabolism, honestly, there's always some individual variation that's occurring, but it wasn't very much. They really all were quite unhappy in those CO2 added pools, and they all had relatively more or higher metabolic rates in that CO2 added treatments. Yeah.
And across like life stages?
I didn't look at life stages. I was just looking at adults within the mussels. That would have been really cool to do, but we were just like, we can see them and they're there where I can collect them across 6 months. So yeah, definitely something we know in mussels and other larval life cycles that, the larval piece is what's that critical life window, right? So I would imagine that regardless of what treatment they'd be in, if you have CO2 or low pH or high temperature, that we would see, decreases in survival in larval forms. I think there was one more. No? Okay. Sorry, Tyrone?
Probably, sorry,
I said Tyrone.
probably know the answer to this, but in the first experiment, did you consider manipulating the muscles so that they were equal in number of clusters?
Yeah, we talked about it, but that might be something that we can do moving forward. Alaska Fish and Wildlife didn't want us doing any transplants. But one, we also just wanted to look at what does the composition look like in this natural system. We didn't want to start manipulating the environment that way yet while doing these manipulations. And then the next summer we went back, which unfortunately was during COVID, so we didn't get to do what we really wanted, but we were gonna start trying to manipulate the community, the mobile community within the pools, and kind of see how biodiversity shifted. So maybe something in the future that I think would be really interesting to see, so we could get more of a, you know, not having pools that have just randomly had a lot higher mussel abundance. Yeah. Do we have time for one more? Yes,
Let's do one more.
okay, one more.
Question, what sort of understanding is there of the genetic footprint of physiological processes that are so important to these species of classification? How much variation is there?
Yeah, it varies a lot by species, is my understanding. And for mussels in particular, I'm not sure we've really looked at that too much, but thinking just across like genetic structures of mussel populations, we know that there's not a lot of local adaptation taking place because they're broadcast spawners. And so you actually get movement of larvae pretty far along the coastline. And so it's absolutely something that we could look at and try and incorporate into future studies of, you know, I don't really know what that looks like for calcification of mussels, but I know that it's an important component. And if you have die-offs of certain genes, right, or certain individuals that are carrying certain genes, you know, how is that going to restructure the community moving forward? Yeah, good question.
All right, well, we have exceeded the 4:30 mark, so any further questions, let's ask them out at the social hour, and thank you all for coming.